So I just want to start by, so we started this thing about 2 months ago. So we raised about $17,000, which is actually pretty good. And so we can actually start doing something. And what we will be doing at this point is that, do you actually see the whole screen? Because I have green box in a very strange place.
We see a green box, but all the slides, presumably.
I mean, do you see the full real estate of the slide?
Presumably, we wouldn't know if we were missing it, but it looks like a complete slide.
I mean, if you can read the complete sentences, then you're good. Okay.
Yes.
Yeah, so this is a joint initiative between Cold Spring Harbor and us. What we started doing yesterday is that we opened applications for the scholarship. So we're looking for graduate students who we will support to attend the Cold Spring Harbor Biological Data Science. And so this is linked from us. This is also linked from them. So essentially, we will be collecting applications until August 1st. This is a fairly straightforward application process. We just ask for 1,000 character summary. We are still in the process of establishing the review panel, but we already have a number of people who said yes, so we'll probably finalize it by the end of this week. And so the logic here is that we're going to collect applications by August 1st. We're going to review them within a week. We will announce winners on August 8th. The deadline for registration is August 13th, so we have a direct channel with Cold Spring Harbor. Basically, we're going to pay for those people that we will select. It's, of course, unfortunately, it's a virtual conference this year. I mean, one of the, I guess, pluses of that is that there is no travel, so anybody can attend that, but it's relatively inexpensive for that reason. So it's also a good sort of test for us to see how it is, but we're gonna, at least with the biological data science, to plan to run it every year. And hopefully next year it will not be virtual, and then we'll expand to more conferences. And we will, in the fall, we will run another, whatever they call this, effort to raise more money. But we have planned to do this right now. So this is, it's really a lot of things here goes to Mike Schatz, because he's obviously, this is his conference. And so he really put us in touch with Cold Spring Harbor, and this has started.
Anton, there's no reason to thank me. This is a really important and meaningful event, and I really applaud you for leading the effort to set up the JTech.
Yeah, I think it's gonna work out fine. Yeah, I've never done this before, so I don't know, but we'll see. I don't think anybody did this before, really. So, okay. So back in February, we started this effort by figuring out how we can use Galaxy platform to help with analysis of SARS-CoV-2 data. Back in February, it seemed relatively benign. And I just started looking for some data, and to my horror, I discovered that actually there's essentially no data. So this was easy to do this first thing. And then wow, that's a horrible resolution. But anyway, so when we quickly wrote this thing, which got accepted fully yesterday, it was just horrible back and forth with plus pathogens, but it's out. I mean, it's not out yet, but it will be out within days there. It's interesting how we always knew that the review process takes time, but this time it's especially profound because this paper basically is about 4 SRA data sets. And of course, it took us about, 4 months to get it through review. So now you have 20,000. They're not really much better than these 4, but it just tells you how this general publication machinery is falling behind the reality on the ground. But the point of this paper was not really to shine. So the point of this paper was really to emphasize the fact that even with the current urgency, even with the current worldwide effort, we are falling very short on doing this analysis in sort of a responsible, reproducible, and open way. So just for those of you, I think I was showing this picture in some of the meetings already. This is just a short reminder of what this beautiful virus is. It's aside from the fact that it causes bad disease, it's a beautiful machinery. It's basically one very long transcript, and it has a number of open reading frames. It has two very large reading frames at the 5' end. They essentially cover about 2/3 of the virus. And then it has, so these big open reading frames code for so-called non-structural proteins. And then towards the 1/3 at the 3' end, you have structural proteins. And it generates this variety of RNAs. So these are you can have full-length RNA, plus you can have these subgenomic RNAs. And since usually only the first open reading frame is actually translated into proteins, so for example, this subgenomic RNA will express spike protein, and this would express 3A, 3B, and so on. So it's a very interesting system with this discontinuous transcription, because all of these short RNAs, they still have the same cap. So they have the same 5' region, which is getting attached to this partial transcripts because of these internal homology regions. And these large products, which are generated from the upstream 2/3, they get cleaved by the proteases that the virus encodes itself. And one of the distinctive features of this thing, which also makes it so large, because at 30,000 base pairs, this is probably one of the largest RNA viruses. This is positive strand RNA virus. So it has this exonuclease, which, so the unique thing about this virus is that it has proofreading activity, because RNA viruses in general are not very good in copying themselves, but this virus has a way of monitoring the quality of replication, and therefore it actually doesn't mutate that much. And I'm going to show you an interesting paper from Michael Lynch, which talks about effects of that. Well, actually, it talks about what would happen if we somehow disrupt that ability of the virus to do this quality control.
Is that actually, is that COV2? Because, sorry, I was just curious, is that actually COV2 since it's a
it's dated
2015
2015,
graphic?
right?
This is not COV2.
This
particular paper is not COV2, but COV2 is the same structurally.
I see. That's, yeah, that's okay.
So there are small differences. This is the original SARS back from 2002.
You have not uncovered the Illuminati, Nate.
Sorry?
Nate has not uncovered the conspiracy.
Oh yes, no. Oh yes. Well, yes. You know, Bill Gates knew all along. Anyway, so
This is being recorded.
I know. Shit. Okay. So SARS, MERS, SARS-CoV-2, they look the same. They do have differences, but the general structure is the same. They all have proofreading activity. So this is the state of the data as of today. It's this number, actually, somehow, when I looked at it 3 weeks ago, it was larger. So I need to check what's going on with the metadata. But roughly we have, you know, between 20 and 30,000 raw read data sets. Those mostly come from Illumina and Oxford Nanopore. There are some data sets from Beijing, from the BGI sequencer. And there are 2 data sets from PegBio as of today. So PegBio is not totally abandoned by this effort. Most of the Oxford Nanopore data is processed. So you can only get FASTQ files. You cannot get the original files. One of the reasoning for that is when they do metatranscriptomic sequencing from patients, they do not want to share the human data Of course, there is a way to get human data out from FAST5 files, but many places don't know about that. So in general, there is no access to raw data as a rule. So
Anton, you're missing actually the main thing.
yes.
SRA.
Sorry, what?
SRA. Good luck loading FAST5 files.
So SRA is, you know, we're being recorded. So anyway.
And you can upload the FAST5 to SRA.
Yes.
It's just, you can't download it, but whatever.
Well, the challenge is that, again, back in the February and in March, when we were trying to get, so there were a few places which were generating superb data, and we asked them to give us FAST5 files. They just cannot do it because there's human data there. So yes, there are two levels to that. First, there is no established way to actually pruning the human data out, and of course it's difficult to get FAST5s actually distributed. And there is still this inequality between the number of complete genomes and the number of raw data sets. So GisAid collects the complete genomes in a semi-open way, and there is no way to know how they were assembled, where the raw data is. And one of our co-conspirators in this effort, Sergei Kazakovsky-Pond at Temple, so he's actually doing essentially daily reanalysis of GisAid genomes. And there is all sorts of random stuff there. There are very bad assemblies. There is also, there is no versioning. So on multiple occasions, something gets deposited to GisAid and then it gets modified later, but there is no record of that. So they only know this because they make snapshots and sometimes they compare, but they actually have all that data.
GisAid will also change your metadata without notifying you. This
exactly. There is no versioning of any kind there. So you have to, all has to be taken with a grain of salt. So, and this is, so in this manuscript, which has got accepted, that's the quote, that's the part of this manuscript which was written by Andrew Loney. I think that's basically our manifesto here. That's what we're trying to do. But I don't think we're gonna get there, at least not during this pandemic. Okay, so our goals were, well, back in February, I guess there was not that much data. So one of our initial goals was to develop a series of workflows that will serve as a kind of best practices and also show that, you know, Galaxy is a big thing across multiple sites here in Europe, in Australia. We wanted to show that we're in sync and you can do analysis anywhere. You can do the same analysis on any site. So regardless whether you have access to infrastructure or you don't, you can analyze this data for free using open taxpayer provided infrastructure. And I think we succeeded in that. And I am eternally grateful to EU folks in particular because they really took on this effort. And I think basically, you know, when I getting bogged down, I talk to them and they inspire me to get back on that. So this is fantastic. And you know, Bjorn, Wolfgang, Milada. So thank you very much for pushing us further on this one. Okay, so we wanted to create a portal for the best practices. We're still in the process of thinking what this should be. I think I was probably rushing it and sort of stressing myself for no good reason, but eventually it's really working out. So what this is, it's essentially this website, and on this website we are providing access to the workflows, to the resulting data sets, and to exact outlines of how we how all these analyses are done. So the main directions are genomics and evolution, and really it's, you know, genomics and evolution and Arctic sequencing, it's kind of a one thing. Then there is a direct RNA-seq effort, which is led by Freiburg. They are generating data, and also chemoinformatics and proteomics, also led by Freiburg. So for genomics, this is started with genomics effort. Our initial goals were to see if there is variation within host. So are there different viral haplotypes inside a single individual? And there was some early evidence for that, for example, the upper and lower respiratory tract might have different viral, not subtypes, but just different haplotypes. Again, there is evidence for multiple infections, and in general, what's the substitution dynamics? How does it change? And SARS-CoV-2 data, primarily to the vast majority of the data obtained with two methods. It's either RNA-seq or amplicon sequencing. Actually, I really listed them in the incorrect order. Most of the data is amplicon sequencing, and about maybe 25% of the data is RNA-seq. So the difference between these two is that, when you are dealing with human samples, when you take a swab, there's actually not that much virus there. So one way to make sure that you're not paying for sequencing human or some other DNA, you actually want to enrich your sample in terms of viral sequences. And so you're doing this by amplifying the viral genome before you create the sequencing library. And places like, for example, the Arctic network effort, they generated really nice styling sets of primer pairs, which allow you to cover the entire genome. One problem with that, of course, is that you're amplifying before you're creating a library, and amplification always has biases. So it's not quite clear, for example, whether you can use amplicon sequencing to assess how variable virus in a particular individual is. Although, of course, the Arctic network people have published on that. And so they think that it's pretty good for Illumina data, not so good for Oxford Nanopore data. Oxford Nanopore data can sort of validate that you have perhaps that particular haplotype, but it doesn't really tell you what the frequency of this is. But this is expected. And the second type of data is metatranscriptomic data, which is when you take a human sample, for example, the liquid from lungs, you isolate total RNA from that, you perform reverse transcription, and you generate library from that, so you don't enrich. And that gives you a much better, so for example, if you're looking at, again, to answer questions, are there variants within a particular patient? That gives you a much better idea, but you're sort of wasting a lot of sequencing on sequencing some of the human RNA, also sequencing, because sometimes COVID-19 patients also have other infections, so co-infections, bacterial infections, so you're sequencing that too. In fact, some of the samples that we analyzed, they have some of the classical bacterial contaminants, which you would expect in patients which show pneumonia symptoms. So what workflows do we have? We have, the first workflow we developed was for variant analysis from RNA-seq data. Then the Wolfgang led the development of variant analysis from amplicon data. This is a combination of Arctic methodology where we just dropped in a different variant caller because their variant caller approach is somewhat primitive. Then we're still thinking about what to do with Oxford Nanopore data. In particular, again, there is no raw data, but again, Milad and Nate were thinking about how we can covariance once we would have that data. And Freiburg folks generated direct RNA sequencing data, and they also created a robust workflow for analysis of this data, which also allows to find, for example, the nucleotide modifications. So, and it's in progress. So that's basically 3 stages. Some are ready, some are in testing, and some are we're thinking about. This is probably about a month old. This is, so initially what we wanted to do, and Marius will talk about this in a few minutes, we basically wanted to grab data daily and analyze it daily. And initially this was doable, but the problem, probably about a month ago or maybe 2-3 weeks ago, we realized that it really doesn't maybe make sense to do this for all the data sets, because as with any research effort, a lot of data sets that are being submitted are simply bad. So, and plus you really need to treat metatranscriptomic data and ampliconic data differently. So it's more fine-grained than that. So I think our strategy from now, we're still doing this. So Marius's automation pipeline still pulls from SRA once there are new accessions, and it does pre-processing and variant calling. But we are thinking about how we're going to publish this. And I think what we're going to do is we're going to concentrate on a high quality subset of metatranscriptomic and ampliconic data to just show A, that our pipelines work, and B, that we can find interesting stuff. And this map shows what we mean by interesting stuff. So the shape of the marker here tells you what kind of mutation that is. So if it's a triangle, it's a missense mutation, means it's a mutation that changes codon, so it's changing underlying amino acid. There are also some crosses here, which really should not be happening, and these are nonsense mutations. These are mutations which create stop codons, so they generate truncated proteins. And in a viral genome like this, which is packed with coding regions, you don't really expect stops. So it's a good proxy for figuring out what the sequencing and in general variant calling false positives might be. But you can see that there are these large triangles. That means that it's a variant, which is in this case, it's fixed because its frequency is approaching 100%. And it's large because it's in many samples. And you can see that there are large but slightly smaller triangles at other sites, and they have intermediate frequencies. So there is a lot of these variants which are polymorphic. They might be fixed in some individuals, they're not fixed in others. So there is something going on here. But in general, the virus doesn't mutate much at all. It's a very stable thing. And so basically we have two sources of data. We have raw reads. So from raw reads, we can see which sites are polymorphic, which sites vary within an individual. On the other hand, we have GisAid data, and that's complete genomes. So from GisAid data, if you pull high quality genomes, you align them, you find variants, you build trees, you see where these variants are in relation to, well, do they change codons or not, whether they have effects like this. You can actually start painting a more full picture of how virus evolves. In particular, what's interesting is that which sites are under positive selection, so which are good for the virus from the viral standpoint. As you will see, there aren't that many of these sites. And one of the reasons for that, of course, is that it's a new infection. It's also, there are no drugs, so there's nothing to evolve out of. Like, for example, in HIV, there is a large set of classical drug resistance mutations. Well, we don't have any drugs for SARS-CoV-2, so there are no drug resistant mutations yet. But obviously, once drugs are developed, then what we're doing here will be extremely useful, because you will be able to use all the same tool set for sort of figuring out how, for example, SARS-CoV-2 avoids certain types of drugs. It will be very interesting from a scientific standpoint. And also, is there evidence of recombination? Do viruses recombine? Is there any evidence for that sort of? You have piece of one virus at 5' end, piece of another virus at 3' end, or somewhere in between. You can, unfortunately, there is really not enough variation in SARS-CoV-2 to do this reliably, but as time goes on, we'll have more data. So this is the kinds of workflows that we currently have. So this is, again, in collaboration with a Temple group, with a DataMonkey Hi-Fi group. So analysis of episodic selection at certain branches in the tree, analysis of pervasive selection, and analysis of recombination. We have all these workflows already done. One of the people who worked on that was Nick Kinnear in my lab. And Temple Group puts out this dashboard. So they generate various kinds of data. And so in this case, for example, they rank all variable sites. They rank them by sort of what's the likelihood that a given site is under selection. And so far, the top, there aren't that, maybe there are, I think the current numbers, there are 5 sites which are really interesting, just 5 sites so far, just 5 sites, not much more really. And the top site here is this particular site in RNA-dependent RNA polymerase. So it's ranked so high because there are multiple lines of evidence. Its frequency is increasing. It's also overlaps with data which we generate from raw reads. This site is frequently polymorphic. So, and there is documentation tab provides various sort of tiers how the Temple group ranks sites. And so this particular site is the top one. And this dashboard, you can go through all the sites, see how its frequency changes. It's for different builds of GisAid. So it's interesting if somebody is working and they find a particular site, they can go here and look at that particular site, but you can also download all this data and analyze it in a more programmatic way when you're not interested in just one site, but you're interested in global patterns. And these 3 workflows, what Galaxy does, what it allows you to do is that you start with a set of genomes. Then the workflow generates codon alignments for particular open reading frames, and then it performs the analysis, selection analysis, or combination analysis on these aligned reading frames. And in the end, it actually generates a visualization like this inside Galaxy. So the site that I was showing you, it's a static site that they maintain as the new data becomes available. But what we're going to have in Galaxy is that if you have a set of complete genomes, for example, for SARS-CoV-2, you can just load them and generate such dashboard for yourself. It's the output of these. The output of these 3 workflows gets combined and into a dashboard like this. So this is an interesting paper, which I actually found just last week, although it's been around for a while. It's been published in April. I don't know if you know who Michael Lynch is, then you probably know this. Michael Lynch is the guy who published Genome Architecture, a very nice book. And well, he's known for many things. He's the Academy member currently located in Arizona State University. So the idea of this paper, it's interesting because it has direct implications with the types of things that we're trying to find. Is that one of the strategies for perhaps fighting with SARS-CoV-2 is somehow kick it off the balance in terms of destroying its ability to keep replication errors in check. Because if, for example, if we target proofreading activity specifically, it's interesting that all the drugs that are developed, that are being developed, well, most of the drugs that are being developed right now, they're targeting either main protease or RNA-dependent RNA polymerase. But what about the, for example, exonuclease, because it's responsible for proofreading activity. So if we can throw this off balance, then if we can find drugs which target that particular activity, it will lead to mutational meltdown, because most of the mutations in any systems are always deleterious. You have to be lucky to find a beneficial mutation. So if mutation rate increases, then this will it will kill virus on its own. That's one of the take-home messages of this very short note. I already showed the portal. This is the old view of the portal. And so I think
Anton,
yeah.
is there any specific locations you're trying to see the mutation load specifically affecting the pathogenicity?
Well, if you if you somehow turn off the proofreading activity, I mean, it's going to mutate everywhere. But
right, but many other regions may not be such effective for decreasing pathogenicity.
That's a shotgun effect. You really don't need to target anything. As long as you increase the mutation rate, it will be, well, to a certain extent, of course, then you'll achieve the goal.
Okay, thank you.
It's much more difficult to target specific regions. But, if you look at the genetic code, most of the changes that you would do, they're probably going to be non-synonymous. So they'll be deleterious by definition. So I heard that joke, I think, from Ennis, is that, the COVID-19 is really the biggest vehicle of progress. For example, we never knew that we can use Zoom for everything. And we obviously never knew that we can actually work from home. Well, we tend to go crazy, but in fact, it can be done. There is no real need in meetings. Sorry?
Some of us knew.
Yeah, some of us knew, yes, but little did we know. So, so actually, it also provided an interesting mechanism for improving Galaxy. So once we started pooling this data from SRA regularly, we started to have millions of data sets under a particular user in Galaxy. And that really, I mean, we knew that Galaxy UI has issues. We're working on resolving these issues. But it really showed that these issues can be quite severe when you're getting to that amount of data. So it's an excellent testing strategy. So we do need to scale the UI. We need to be more robust towards failures, because once you pull 5,000 short-read archive data set from SRA and some of them fail during the processing, and what are you going to do? Run all 5,000? We do need to think more about good genome browser integration. And so actually should we revive Trackster? Should we use HighGlass? Should we invest in IGVGS? We really need to solve this problem. Another problem, which is not really Galaxy related, is the refactoring portal, because we want people to get to our results and workflows more easily without much reading. And I think it also renewed the need for this scaling, syncing across main Galaxy instances worldwide. So, Marius, you want to take it from here?
Yeah, sure.
Should I stop sharing, or just tell me? You're gonna just.
Yeah, I will just tell you when to go to the next slide.
All right.
So now it's not too uncommon that we generate around 100,000 data sets per history. I mean, we actually usually still. Can you stay on that slide? So we are approaching 100,000 data sets per history, and that's kind of the challenge. So first, when you load the history, it's still kind of acceptable. Loads within like 2-3 seconds, while additional information is fetched in the background. But once you open a collection that has maybe 5,000 data sets, that takes a while because everything is fetched at once. So something else that we do is whatever we download from the SRA, we also put in the library, so users can take individual data sets out of the library. If in fact we could open the library before it turns out. So fortunately, Oleg from the EU team has already started working on it. But I mean, that's one thing that became crystal clear. These histories, when they have 80,000 data sets, they are importable, but it takes like, yeah, about 20, 30 minutes while it appears that nothing is happening, and then you see a gateway timeout. So there are many small things where, I mean, it's fine that this sort of thing takes a while because it's just a lot of data. But where we need like smaller improvements saying that, hey, Galaxy is actually doing stuff and you don't need to do it again, because if you do it again, it will just take even longer. There are some things that worry me a little bit more, like for instance, we have a dataset search that I use a lot that I only discovered some time ago. So if you don't know it, you can go to the user menu and datasets, and you can search for your data sets, and the search is really good and it's already quite optimized. And yet querying, I mean, just loading up the first page can take like 3 minutes, which, I mean, again, it looks like nothing happens, but also we need to be able to start searching before that. In the same way, we have this menu where you can see your different histories and how many data sets are in there. Again, these are already optimized queries that just count the number of elements in the history, but that also takes like 2-3 minutes to load. And I expect it won't take long before this doesn't load at all anymore and you just get a gateway timeout. Yeah, I mean, some other things are being worked on. So I guess multi-history views don't be revised soon, but it's basically not usable. Once you click there, close the tab, try again. Similar, yeah, I mentioned the history import, the history export, it's the same story. Again, you click on export. Nothing happens for a while and you see gateway timeout. Another thing is collection and workflow scheduling. So I've started working on it. There are many places. So in principle, if you're not familiar with this, like the collections, they contain individual data sets, and a lot of it can be done uniformly and just once. But in some cases, we do something for each individual element in the collection, for each element in the collection. So we quadratically increase in time with every element in the collection. So these are not super hard to eliminate, but it takes some effort. And whenever you start working with these sort of things, it's good to keep an eye on performance and whether you might be limiting yourself there. So, okay, next.
So this dataset search bullet, is it about the search under user datasets or in history?
No, the history search works okay.
Gotcha.
Well, I mean, the pagination is painful when you have to go to 20 pages, but it works.
Okay.
Okay, so this is the numbers of data sets that we're speaking about. So these are the input data sets. And when you run this kind of analysis workflow, each data set until you're at the end produces like 30 data sets. So that is really a lot. Yeah, I think that's all to say there. So the largest deposition on a single day was like 8,000 data sets. And that took like 4 days to run on usegalaxy.org, which is not terrible, but it could be faster. And for the most part, it's not actually limited in terms of cluster capacity, but how fast we schedule the jobs. So we should make sure that Galaxy scales at least in a way that we can saturate the computational resources we have before like job scheduling becomes a problem. Which is absolutely feasible. I think we can do that. Okay, next. Yeah, so this is just the mechanics of how we do it. So we have a GitHub workflow that queries the ENA API at this point because it's slightly easier to use than the SRA and has less marks. and creates a pull request whenever there are new data sets. And then the second part is we also launch a workflow with the new data sets that have been found. So that, yeah. Okay, so we have this is the basic workflow. So this workflow that does two things, so the single and the paired end alignment. And it would be easy to add additional things like nanocore analysis and splitting the Illumina data sets in amplicon or not, except we don't have the metadata to do this properly. But yeah, okay, next. So the point is there's also many ways to create Galaxy workflows, and some are better than others. So one thing is we have this possibility to have typed inputs, so meaning we have our data sets, but we also have things like text fields, integer fields, float fields, booleans, and so on. You can set them either with an old style parameter, or you can use new things that we have. If you use the new things, these workflows describe themselves. So what you see in the interface, you can easily reproduce on the command line. I've written a small utility that just takes the workflow ID, takes the Galaxy instance, and will create a script. That script will run the workflow using BioBlend. So initially I just wrote it because people, it was a bit complicated to figure out how to launch workflows with BioBlend, especially if you were using these new style parameters. But that utility just templates out the actual script. And that templating, I think we can also use in Planemo to make it easier to actually generate all of the test files that you need to have in order to properly test workflows like we test tools. Okay, so yeah, next slide. Yeah, so that's on the left is the published history page, so you can see all the histories that have been published. So we started the first run of the automation was May 3rd, and the last update was like 3 days ago, which was the last big data dump. One of these histories there with 80,000 data sets, so that took 2.5 days. So that is also after some tuning of settings that we've done with Nate, so this is a bit faster than it used to be. Okay, next slide. So we sort of solved the problem of how to run these workflows automatically, but we also want to update the portal with the results. And I think one cool thing that we could work towards too, and I started doing a bit of this, is that instead of just coming from the outset and triggering workflows, triggering data analysis in Galaxy, it would be good if Galaxy could also be like this universal workhorse that is there on the web, always available, always accessible, where you can do long-running jobs instead of polling, asking, is the Galaxy server already done, which is something we can't really do in a CI system. Instead, Galaxy would say, when certain events occurred, would send out a whatever post request with information about the job. So I sketched out something here, and I wrote a corresponding GitHub application that can verify that a request came from a given Galaxy instance by a given Galaxy user. And you can install this application on the GitHub repository, and then the application can post comments on behalf, on that repository, and you can take actions based on the comment. So this sort of closes the loop, and what you could do, for instance, with this is take the results of the workflow, accumulate them with all the previous results, run them through an IPython notebook or whatever within a GitHub action, and then update the home page with the latest variants, the latest numbers, and so on. So on the Galaxy side, this involves being able to verify, to hand out and verify JSON Web Tokens. But other than that, I think in general, it would be a good idea if Galaxy can say, this event occurred, and I will send it to whatever place, and that place can do something with that information. Yeah, that's it.
Thanks, Mario. So I'm just going to quickly mention some of the other sub-projects that are happening. So Nick Kinnear, in addition to writing this workflow, which does the selection and recommendation analysis and generates the dashboard that I was showing, he wrote a little tool which does codon alignments. It's surprising that this problem, I mean, it's a conceptually straightforward problem. Given, you know, a set of DNA sequences, produce a multiple alignment which is codon based. So essentially meaning that all of your gaps are in triplicates and between codons. But in reality, there's actually no tool which would do this sort of effectively for, so Marius, you're controlling my screen, I think somehow. So I gave you that opportunity, so use it wisely.
Nice.
So basically,
salary raise for everyone.
So Nick wrote a utility that can, for example, you can give it a 10,000 SARS-CoV-2 genomes and tell, give me all codon alignments for spike protein, and it will just do it. And it's just fantastic. So we are writing an application note on that. It's bottlenecked at me. I need to finish editing this. And if I may, I'll get out of all of my meetings today and actually do this. So we'll see. Then, of course, there is the Data Monkey portal, which I was showing you. So Han is working on the interesting problem is that in SARS-CoV-2, you have ORF1A and you have ORF1B, and it's essentially one thing. So you have an overlap. And in order to start translating, in order to switch frames, what happens is that ribosomes sort of slip one nucleotide back. And that happens because of the particular secondary structure in that region. It's always fascinating how things like this evolve, because there's a lot of constraints here. First of all, you need to keep that secondary structure. Second, you need to make sure that your coding region overlap is preserved. So it's really interesting. And the amount of overlap is almost like a, it's like a phylogenetic character. You can classify viruses based on the structure of that overlap. For example, you can classify alpha, beta, gamma coronaviruses based on that alone. And so Han was looking across all viruses with ORF1A and ORF1B, what he can find, and figuring out what was the ancestral state, how it's happening. This is another manuscript which is in the works. So he's done a lot of work. This actually shows the different delta, alpha, and gamma coronaviruses, and this is amount of overlaps between the ORF1A and ORF1B. So also Delphine was working on, one of the, again, it's almost like the problem with the generating codon alignments is that when you have a new virus like SARS-CoV-2, I mean, we've been studying coronaviruses for quite some time. So there is information, for example, about particular amino acids in SARS, in original SARS. There is information about particular amino acids in MERS, in the infectious bronchitis virus, in pig coronaviruses and so on. But of course, it's a shifting coordinate system because they all have different lengths. So for example, I read a paper and it's about infectious bronchitis virus, and it tells me that this amino acid is important. So the natural question is that what's the corresponding amino acid in SARS-CoV-2? And so she generated this notebook which allows you to do this coordinate conversion easily. And she also did painstaking work of looking through the literature and basically associating individual papers with all residues in SARS-CoV-2, mapping it across. So this is a really valuable resource. And so we also have a note, writing up note in process about this. And she's in doing that, she also adopted observable QC, which is an interesting notebook system. If you like doing data analysis in JavaScript, it's all yours. And I'm just going to finish on, there is a direct RNA sequencing data out of Freiburg, and we'll be seeing a lot of interesting results from there. And it's going to be fully functional Galaxy pipeline for analysis of raw direct RNA sequencing data. I'm going to just stop. So if you guys have any questions.
I have one for Marius. You mentioned something about BioBlend it's complicated to invoke workflows, right? Is anything we could add to BioBlend to make it simpler?
I mean, we already added the inputs by name. I think that's the key, and maybe we should make that the default.
So it's,
previously, you needed to figure out what is the ID of the input, right?
Well, we don't need to discuss it here. Or just yeah, maybe open an issue or something that we could track and see what we could improve.
I like Vijay. It's windy where you sit. It's interesting. Don't get blown over.
Yeah. No tsunamis.
Okay, so that's a short report of what we're doing. So our biggest goal, I think, in the future is to figure out to actually make Portal really, really useful for the. And again, I was worrying that we're not going to get it in time, but I think that we have time to do it well. We don't really need to rush this to this extent. SARS-CoV-2 is going to be around for a while, unfortunately.
Marius, where's the repo that the workflow runner repo? What's the name of that?
You mean the GitHub app, or how it's integrated in? That's a good. I mean, it's not ready, right? It's. I have to check that out. I don't remember. It's not ready in any way, right? Just see. I will send it in a second.
All right, thank you.
And where's the GitHub action running from for the current runs?
That's within the SARS-CoV-2
right. Yep.
repo.
Okay. So BCC starts next week. So we're going to be busy. So as John said, no roundtable in 2 weeks.
All right. Thank you, everyone.
Thank you.
Yeah, thanks.
Everyone.